Hello again, students, and welcome to Lecture 11 of our series on human biology. This lecture we will be discussing the renal system, which is the kidneys. It's also known as the excretory system or the urinary system. And in humans, humans are land-dwelling mammals. And so for humans, the main purpose of the kidney system, the renal system, is to filter poisons out of the blood, particularly ammonia wastes and some other waste products that need to be filtered out, some other dirty stuff in the blood that will not leave through the lungs and is not filtered out by the kidneys, by the liver, for instance. And mostly we're referring to ammonia. When you break down proteins, sometimes you accidentally break apart some of the amino acids. We know that amino acids have lots of nitrogens in them. That's part of the reason why they're called amino acids, because they contain amino groups. If you break an amino group off of an amino acid, you end up creating ammonia, which is a very toxic substance, and we need to get rid of it. So there are different strategies that different animals use to get rid of this stuff. If you're a fish, you live in the ocean, you can basically, the ammonia will come out of your gills into the water directly, so you don't have to worry about it too much unless you want to. If you're a land animal, you have to worry about how long you're going to retain the poison in your blood before you get rid of it. And in the case of, if you're going to retain it for a while, for example, you've probably heard the expression that animals don't like to urinate where they sleep. So that means that if you're sleeping, or if you have a place where you live, a hole at the base of a tree or a nest or something like that, or in a tree or a house if you're a human, you don't want to be urinating everywhere. You want to go someplace, urinate, leave the urine at that other place, and then go back to where you live. So that means you have to retain these poisons for a little while. So it may be in your best interests to convert it into a less toxic form than ammonia. Ammonia is very toxic. You might be interested, if you have to hang on to these ammonia waste products for a certain amount of time, it may be in your best interests to convert it to a less toxic form. And that will probably cost you a little bit of energy, which it does, but it's worth it. So mammals convert ammonia in their blood into a slightly less toxic compound called urea. This takes place in the mitochondria. You remember what mitochondria are? Those are cell organelles that we talked about in the first week. And in the mitochondria of liver cells, ammonia is converted into urea. And that is the name for that process is called the urea cycle. And it takes place, it's a cycle. Whenever you hear the word cycle, you can assume that we're talking about a series of chemical reactions that kind of, at the end, you end up with some of the things you started out with, so you call that a cycle. So we talked about the Krebs cycle that happens in mitochondria. That's the conversion of glucose into ATP. And another thing happens in the mitochondria, which specifically the mitochondria of the liver, which is that ammonia is converted to urea, and that is called the urea cycle, and it takes place in the liver. So when you break down food and when you eat food, you eat the food and you break it down, you end up generating a lot of ammonia. That ammonia would be toxic if it was put directly into the blood, but you now know that it doesn't go directly into the blood because the nutrients that are absorbed from the intestine are sent into the hepatic portal system, and then into the hepatic portal vein, and then into the liver, where they are detoxified. And so one of the things that the liver does is that when you eat food and you have this ammonia in the food, it gets sent to the liver. The mitochondria in the liver will convert it to urea, which is less toxic. Okay, so that's one of the ways that the liver filters poisons out of the food we eat. And there are other ways, but we're not going to go into it in this course. So once the liver has converted the ammonia into urea, it's free to circulate around in the blood for a little while longer, and then it is eventually filtered out by the kidneys and concentrated into something that we call urine, and then sent out of the body as a waste product. Okay, so that's the main purpose of the human kidney system, but I also wanted in this lecture just to touch on some of the other things that other animals do with their ammonia waste products. Fish that live in the ocean generally just dump the urea out into the ocean because they don't, they're going to move on anyway, they're going to swim away anyway, so what do you care if there's a little bit of a higher concentration of ammonia in a certain part of the water. All right, so let's take a closer look at the renal system. Okay, so the original purpose of the renal system, if you're talking about fish, remember that animal life evolved in the ocean before it moved on to land. At least that's what the evolutionary biologists believe. So the original purpose of the renal system was not only to get rid of ammonia, but also to regulate the amount of water that's inside sea animals. So at the very beginning of the course, we talked about chemistry, we talked about a concentrated solution versus a dilute solution, and what happens to cells, what happens to living cells if you put them into a solution that is either hypertonic. Hypertonic means that it has a higher concentration of solutes than inside the cell, or if you put that cell into a hypotonic solution. A hypo, the word hypo means below or less than. The word hyper means above or more than. So if you put a cell into a hypotonic solution, water will go into the cell and it will swell. If you put a cell into a hypertonic solution, water will go out of the cell and the cell will shrink. Okay, so if you're a fish and you live in the water and the ocean has a higher salt concentration than your cells do, then you have a problem. It means that you will shrink, you will lose water. And in fact, if a human is marooned in the water, let's say you were in a shipwreck, and this actually happens, unfortunately, if you find yourself floating around the ocean wearing a life jacket for days, you will actually die of dehydration, because the ocean has a higher tonicity than the human body. And so that means the ocean has more solutes dissolved in it, which means that the fresh water that's in the cells of the human body will diffuse out of the human body into the ocean in a vain attempt to try and equalize the amount of salt on either side of the membrane. That's actually what your body is doing. So in fact, if you float around in the ocean for too long, you'll dehydrate and die from dehydration. If you drink salt water, you've seen shipwrecked sailors, you've heard of shipwrecked sailors that have no water. They end up drinking the ocean water and they end up dying of thirst, essentially, because the ocean water sucks there's so much salt that it sucks the water out of the cells. All right, so the original purpose of the kidneys in fish, the renal system in fish, was to, and I'll explain how this works, the original system was to get rid of as much water or as much dissolved compounds like urea or ammonia, get rid of just enough of those compounds to balance the osmolarity of inside your body versus the water that you live in. So one of the original purposes of the renal system was called osmoregulation. Osmo refers to osmolarity, osmosis, and then osmoregulation means you do various things with your blood, the amount of liquid versus salt in your blood, in order to keep you from losing water or gaining water if you're hypertonic or hypotonic relative to the solution that you're living in. If you're a freshwater fish, you have different problems than an ocean water fish. Second purpose of the renal system is to eliminate nitrogenous wastes. Of course, nitrogenous wastes contain nitrogen. If you break nitrogen out of whatever compound it was in, either an amino acid or a nucleotide or a base for a nucleotide, all of those things contain nitrogens. If you break the nitrogen out of those things, it tends to turn into ammonia, which is very toxic. And so we either get rid of the ammonia right away or we convert it into a less toxic form and get rid of it later. Okay, so then once we've discussed that, then we're going to discuss the gross anatomy of the human renal system. And there's a difference between gross anatomy and microscopic anatomy, by the way. So gross anatomy refers to the level of organs that you can see with the naked eye. So you don't need to look at things under a dissecting microscope to see what's going on. We call that gross anatomy. If you slice things up and look at individual cells under a compound microscope, we call that microscopic anatomy or cellular anatomy or tissue or histology, for instance. Okay, so gross anatomy, we will look at the big picture, the large-scale picture of the human urinary system and all the parts that form it, including the kidneys, the cortex of the kidneys versus the medulla. Remember that the word renal refers to the kidneys. So the renal cortex, I told you in an earlier lecture that the cortex is the term that you use to talk about the outer surface of things. And the medulla is usually what you use to refer to the inner part of an organ. So there is a renal cortex, the outside of the kidney, and there's a renal medulla, which is the deep inside the kidney. Okay, so once we have looked at the gross anatomy of the renal system, the kidneys and the urinary bladder and the ureters, we'll look at the microanatomy or the microscopic anatomy of the human renal system. And if you look inside a kidney and you cut it up into, if you slice it up and then you look at it under high magnification, under a compound microscope, you will see that it's composed of billions and billions of little tiny filters. And those little tiny filters, individually, one of them is called a nephron. And then if you're talking about multiple ones, it's made of billions, the kidney is made of billions of nephridia. So nephron is the singular form, nephridia is the plural form. Now within a nephron, you have a filtering system called a glomerulus, which is located inside a structure called Bowman's capsule. And what that glomerulus does is it filters, it basically pushes all of the blood plasma out into the Bowman's capsule. The glomerulus is really just a fancy name, a specific name for a capillary that's located inside a microscopic bulb shape called Bowman's capsule. And what happens at the glomerulus is, using high blood pressure, the glomerulus will push all of the blood plasma out into the Bowman's capsule. And then when you push a liquid through a filter, the liquid that ends up on the other side of the filter that the liquid that comes out of the filter is called the filtrate. The filtrate. So when you filter the blood through the glomerulus, you keep the cells inside this capillary called the glomerulus, and then you push the serum out of the glomerulus into Bowman's capsule. The serum then goes down and back up again through a loop, a little tiny tube that's in the shape of a loop called the loop of Henle. And as this filtrate, the blood plasma goes down and back up again through the loop of Henle, the water is recovered. So in humans that live on land, and we have a problem, you know, unlike a fish that lives in a freshwater lake or in the ocean, we have a problem getting water. So one of the keys of the renal system, one of the key functions of the renal system is that it has to be able to filter out the urea but take back the water. And that's kind of a trick. And that is mainly what's happening in the nephron. So the blood plasma that contains both salt, urea, and water, that blood plasma is pushed out of the glomerulus into the Bowman's capsule, and then it is sent down and back up again through the loop of Henle. And as that filtrate, that plasma travels down and back up through the loop of Henle, the water is taken back. And then what's left is now concentrated salt and concentrated urea, and that is sent that is collected in the kidney center, which is called the renal cortex, and then it is sent down a tube called a ureter to the urinary bladder. And so in the urinary bladder we have concentrated urine. It's concentrated because we took the water back out of it. So that's how you concentrate things, is by removing the water. So we took the water back out of the filtrate from the Bowman's capsule, and then we let everything else go out. So that's how it works, and we'll talk about that. And then at the final part of the lecture, I'll give you a very brief discussion of how the amount of water that is taken back from the filtrate in the kidneys is controlled by hormones. And we haven't actually gotten to the endocrine system, but when we get to the endocrine system, we'll revisit hormonal control of urination. Okay, so hormonal control of water retention is one of the hormones that's produced in the brain is called antidiuretic hormone, which is abbreviated ADH. It is also commonly known as vasopressin. So the brain produces a hormone called vasopressin. It's part of the endocrine system, and the vasopressin determines how much water will be retained by the kidneys. So if you produce a lot of vasopressin, the kidneys will suck a lot of the water back out of the filtrate in the Bowman's capsule and the loop of Henle, and also the collecting duct, which is another part of it. And if, on the other hand, if your brain doesn't produce a lot of vasopressin, then you will lose a lot of water and you'll be urinating a lot. So when you get into a situation where you're urinating a lot because your kidneys are not retaining as much water as they're supposed to, first of all, that happens naturally as you get older. You find that as you get older, you have to urinate more often, and that's because the kidneys are not as efficient at removing the water and taking back and reclaiming the water from the filtrate. But it's also because your brain is not producing as much vasopressin. So the less vasopressin you produce in the brain, the more often you have to urinate and the more dilute your urine actually is. Okay, let's take a closer look at osmoregulation. So the original purpose of the kidney system, the renal system in certain types of fish was to maintain osmolarity with the body of water, maintain the osmolarity within their bodies, because it was a concern if you're a fish. If you're living in a body of water, you have to constantly worry about whether that water that you're living in is hypertonic or hypotonic relative to the inside of your own body. If a fish is living in a hypertonic solution, a water body that's hypotonic, that means that the water that they're living in contains more salt dissolved solutes, and water will diffuse out of the fish into the water where they live, the ocean, for instance. On the other hand, if you're in a freshwater lake, generally a fish in a freshwater lake is living in a hypotonic solution, which would normally mean that water would diffuse into the fish and the fish would swell up and burst, which is in fact sort of what happens when you put a saltwater fish into a freshwater fish tank, for instance. It dies fairly quickly because its cells burst because of it went from living in a hypertonic solution into a hypotonic solution. Okay, now there are two, just to back up for a minute, there are two main functions of the renal system. One is osmoregulation. So you're getting rid of waste products, but at the same time, your kidneys, your renal system is deciding how much of the water to take back in order to balance. Also, the renal system not only reclaims water, but it can reclaim a certain amount of the salt ions that are in the plasma as well. So the renal system is reclaiming water and reclaiming a certain amount of salt, just enough to make sure that the osmolarity is balanced with the water that they live in. So we'll talk about some of the problems that are faced by a freshwater fish versus a saltwater fish. We call a saltwater fish a marine fish, or a marine animal. Marine refers to the ocean. Specifically, it refers to the ocean, not to lakes. Okay, now the second purpose of the renal system is to eliminate nitrogenous wastes, either ammonia or urea or other compounds that are derived from ammonia. So when you break down amino acids and nucleotides, for instance, you generate a lot of ammonia in the cells. The cells then dump that ammonia into the bloodstream as a waste product. That blood travels, in the case of humans, it travels through the liver where the ammonia is converted to urea, which is a less toxic version of ammonia. This costs us a little bit of energy to do this. And there are other animals like birds and insects that convert it into an even less toxic substance called uric acid prior to getting rid of it, prior to elimination. So as I said, osmoregulation refers to trying to avoid having the cells in your body shrinking or expanding and possibly bursting. So I mentioned earlier on in the course that if you put a cell or a bunch of cells into a hypertonic solution, that means that the concentration of solutes in the solution is higher than it is in the cells. And so the cell is floating in a hypertonic solution. And that causes water to flow out of the cell into the solution, and the cell shrinks. If that happens to red blood cells, which is actually shown here, what we're looking at here is what happens to blood cells in different concentrations of hypertonic versus hypotonic solutions. And in the hypertonic solution, water will go out of a red blood cell and it will shrink, and you can see these sharp edges on the surface. It no longer looks round, it looks kind of wrinkly. And when that happens to a red blood cell, that's called crenation. I won't ask you that on a test, but that's the technical name for that happening, and it does happen in certain diseases. The red blood cells will undergo crenation because the blood ends up becoming hypertonic relative to the inside of the blood cell. On the other hand, at the opposite extreme, if you have a cell floating in a hypotonic solution, that means that the solution the cell is in has fewer dissolved solutes, and so water will flow from the outside to the inside of the cell. The cell will swell up and possibly even burst. And then finally, isotonic solution is where you have an equal amount of solutes dissolved inside and outside of the cell. So water is flowing into the cell and out of the cell at the same rate. And remember that this is all made possible by selective permeability, which I discussed in lecture 4, I believe, where selective permeability means that water is free to cross back and forth across the cell's plasma membrane, whereas other compounds and other solutes are not free to diffuse across the membrane. So water is free to come and go as it pleases. Other things are not, which could lead to a situation where a cell has a higher concentration of other things, salts, glucose, lipids, and things like that. All of those contribute to the tonicity. Hypertonic, hypotonic, isotonic is referred to as tonicity. Tonic means a solution with something dissolved in it. So tonicity refers to the amount of solutes that are dissolved in the solution. And then we have hypotonic, isotonic, and hypertonic. Okay, so all of these, the tonicity influencing whether a cell shrinks or expands is related to the selective permeability of the plasma membrane of the cell, which allows water to come and go freely. But however many proteins, if you dissolve proteins in water, that contributes to the tonicity of a solution. You can dissolve salts, common salts like sodium chloride, potassium chloride. Dissolved metals will also contribute to the tonicity of a solution. So when I talk about the solutes dissolved in a solution, I'm not necessarily talking about sodium chloride. If I talk about salts, I'm just talking about any molecule that's held together by ionic bonds and which will dissolve in an aqueous solution. So typically, both inside and outside of a cell in the human body, we have sodium chloride, we have potassium chloride, we have glucose. Glucose contributes to the tonicity. We have fructose and sometimes sucrose, which also contribute to the tonicity. Those are all solutes that dissolve in water. Proteins, many proteins are hydrophobic, which means they will not dissolve, but many proteins, particularly the ones that are called globular proteins, they will in fact dissolve in the water and contribute to the tonicity, contribute to the concentration of solutes in that solution. So we're talking about everything that dissolves and not just sodium chloride, for instance. All right, so this is the dilemma that sea animals face. If they are in a hypertonic solution, the ocean is generally hypertonic to most fish, so they have to do something to regulate the amount of water that they lose to the ocean. By contrast, a freshwater fish that lives in a freshwater lake that has practically nothing dissolved in it, they have the opposite problem. They have the danger of too much water going into their cells and causing them to burst. And as I said, that's what happens if you put a freshwater fish into a saltwater aquarium. It will dry out and dehydrate. And the reverse, if you put a saltwater fish into a freshwater aquarium, it will die fairly quickly because it will swell up and some of its cells will burst. All right, now, if you're a fish living in the ocean or in a lake, a freshwater lake, well, first of all, there are two types of fish. Number one are the osmoconformers. So these are generally sea animals, including sharks. This pertains particularly to sharks and dogfish and any of the chondrichthyan fish. These are the most primitive types of fish, and they evolved, and they have not yet formed a renal system. But what they do is um, they take they do have the ability to convert ammonia into urea. And so an osmoconformer is a fish that retains enough urea, and urea, of course, is a solute. So what they do is they convert ammonia to urea, and then they retain enough urea in their tissues in order to balance the amount of salt, other salt in the ocean, so that water there's neither a net gain or a net loss of water into a shark or out of a shark, because it has placed enough urea into its body tissues in order to prevent that. Okay, so an osmoconformer is usually a sea animal that lives in the ocean, in the sea, and it will retain urea in its tissues, just enough urea so that the osmolarity in its body will match the osmolarity in the ocean. The word osmolarity refers to the amount of salt, or the amount of solutes that are dissolved in the aqueous solution. Okay, now you remember when we talked about chemistry, we talked about millimolars and molars, right? So millimolars and solutions in chemistry are usually measured in millimoles, depending on how, did you dissolve 2 moles of salt in a liter of water, then you've got a 2-molar solution, also known as a 2000 millimolar solution. Now, when we switch to biology and we're talking about the amount of salts that are dissolved in cells versus outside of cells, we don't talk about molarity anymore, because we're not talking about a pure solution. We're not talking about a solution of sodium chloride or potassium chloride. We're talking about a solution that is a mixture of sodium chloride, potassium chloride, urea, glucose, proteins, etc. So it's a mixture of different solutes. We don't really care what they are, we only care what the concentration is. So when we're talking about osmolarity, the total amount of solutes that are dissolved inside a cell versus outside a cell, we use a different measurement system. Instead of millimolar, we talk about milliosmoles. Milliosmoles. Right? Now the ocean happens to have a concentration or a tonicity of 1000 milliosmoles. That means that a shark living in the ocean will put 1000 milliosmoles worth of urea into its tissues, so that water will neither go into the shark nor out of the shark as a result of it living in a hypertonic solution. So that is what the osmoconformers do. All right, osmoregulators are another type of fish, and they actually vary the amount of water that goes in or out of their body in order to compensate for the tonicity of the solution that they're living in. So if you're a freshwater fish, water would go, just as an example, a freshwater fish lives in a freshwater lake. It tends to drink very little of the water that it's in, and it excretes a lot. It urinates a lot. So you should bear that in mind when you're drinking the water from a lake, because bearing in mind that the fish that live in the lake are constantly urinating in the lake, but it's usually so little that there aren't that many fish. But if you're in a fish farm, a freshwater fish farm or something like that, then you would have to think twice about the level of urine that the fish are putting out. But generally, a freshwater fish is hypertonic. The body of the freshwater fish is hypertonic relative to the lake that it lives in. And so it is constantly having water diffuse into the fish, and it's constantly swelling. And in order to counterbalance that, a freshwater fish tends to urinate a lot to get rid of the excess water so that it's not swelling up. By contrast, a marine fish that lives in the sea is hypotonic to the ocean. So, it is hypotonic to the ocean, so the water would leak out of a shark if it didn't do something. In the case of the sharks, they put urea into their tissues. In the case of other marine fish, they simply drink a lot of water that they're swimming in, and then they urinate very little, because they already have a problem with water leaking out of the fish. So as a result, to compensate, they urinate very little. Okay, so here we have an osmoconformer. It's a shark. The shark puts converts a bunch of its waste product, ammonia, into urea, and then stores that urea in the tissues, which is one of the reasons why if you eat shark meat, it has a sour taste. I don't advise eating shark meat. Some people eat it as a delicacy, I know, for instance, in Japan they have a delicacy called shark's fin soup, which has it has noodles and shark's fins in it, and the meat from shark's fins. And if you eat that soup, it has kind of a sour taste, a sour, gritty taste, and that is the urea that you're tasting. And urea is a poison, so that's why it's not good to eat shark meat if you can avoid it. There are places where people eat shark meat because it's the only fish they can get. But anyway, so sharks are osmoconformers. They do not attempt, they don't make any attempt to regulate how much water they're urinating or whatever. They simply maintain an osmolarity inside their body that matches the osmolarity of the ocean. Okay, humans and osmolarity. Okay, so I mentioned the fact that the osmolarity of the ocean is 1,000 milliosmoles. Right, so sharks maintain a body osmolarity of 1,000 milliosmoles. Do you know what the osmolarity of the human body is? You can probably guess that it's less than 1,000 because I told you that if humans stay in the ocean long enough, they'll dehydrate. In order for water to diffuse out of the human body, it means that the ocean must have a higher osmolarity than the human body, and in fact it does. So inside the human body, we have an osmolarity of about 300 milliosmoles. So here we have a shark that has an internal body osmolarity of 1,000 milliosmoles, and we have a human that has an internal body osmolarity of 300. So if humans stay in the ocean too long, they dry out. They dry out. So humans, we don't stay in the ocean. That's number one. And if you do stay in the ocean, if you swim a lot, you have to keep hydrated by drinking lots of fresh water to replace the water that diffuses out of the body while you're in the ocean. Okay, let's look at two fish that are osmoregulators. So here we have on the left a marine fish. The marine fish has the problem that water is leaving its body because it is hypotonic relative to the ocean. And so a marine fish drinks lots of water. Right, so it drinks the seawater that it lives in, and it urinates very little. What about a freshwater fish? A freshwater fish has the opposite problem, which is that the water is constantly diffusing into the fish, so it's swelling up. So it drinks very little, and it urinates a lot. So fish that live in the freshwater tend to urinate a lot. If you have a fish tank that has freshwater fish in it, like goldfish, for instance, the easy way to get rid of the urea that the fish are putting out by urinating, the easiest way to get rid of it is to change the water. That's why you always have to change the water in a goldfish bowl. And the more expensive way is to have a carbon-activated charcoal carbon filter that basically sequesters the urea out of the water. It filters the urea out of the water, and then you have to throw away the charcoal because it's become saturated with urea. Okay, what about land animals? Okay, so humans are land animals. We're land mammals, in fact, mammals being a special type of animal. And so our issue is not, water leaking out into the ocean. Our problem is perspiration generally. So if we're living in a hot environment, we perspire, we sweat. The purpose of perspiration is to cool the surface temperature of the skin. So we lose water that way. But we also retain a certain amount of water. We need to have a certain amount of water in our blood in order to carry out the chemical reactions. So humans and other mammals live on land, and so water loss, the skin prevents most of the water that we have from evaporating, so that's not an issue. So the main purpose of the urinary system, the renal system for humans, is to filter out the ammonia and the urea in this case. So that is our main concern. So osmoregulation is not so important for land animals. Water loss is less important. The skin is a fairly impervious barrier to evaporation, so the water doesn't leave. Elimination of ammonia wastes. So we don't want to urinate where we sleep or where we live, so we have to retain the urine for a little while, so it makes sense to store it for a little while. If you're going to store the ammonia waste for a little while, it makes sense to convert it into a less toxic form, which we do. So land animals convert the ammonia into a compound called urea, which is less toxic. This is done in the mitochondria of the liver. and then it is stored filtered out of the blood by the kidneys and then stored in the urinary bladder. Insects and birds convert ammonia into an even less toxic compound that is called uric acid. And because urea and uric acid are both rich in nitrogen, throughout the ages, we live in the modern age where we manufacture fertilizers for farming, but in the old days, 100 years ago, people used to go around the world looking for bird droppings, especially seabirds. Guano is the word for bird droppings. And people used to go and look for tropical islands where there's a lot of bird droppings and put them in bags and sell them to farmers because uric acid was a fantastic fertilizer. It put nitrogen into the soil, which plants need to grow. But anyway, humans don't do that. Humans convert the ammonia into urea, and then we get rid of it in the form of urine. All right, so if a fish is getting rid of its ammonia, there are some fish that will convert it to urea the way sharks do, but a lot of them just dump it out through their gills, right? So some of them have gills, some of them have a separate hole where the ammonia comes out, usually just directly into the water as ammonia. So a fish does not have to worry about urinating where it sleeps because it doesn't sleep anyplace. It keeps on swimming, so it doesn't matter if you put the urea directly into the water, and certainly you're not going to retain it for very long, so there's no reason to waste energy by converting the ammonia into something less toxic. Mammals convert ammonia into urea, and then the urea is filtered out of the blood to make urine, which goes out of the body. Urea is less toxic than ammonia. And then birds and insects and certain mollusks like snails convert the ammonia into a very nice compound called uric acid, which makes a good fertilizer. And so that's what bird droppings were used for thousands of years to fertilize the soil for farmers. In fact, in the 19th century, bird droppings and islands that were covered in bird droppings countries would fight wars over who gets to take the bird droppings. It was called, so you see here on the left there's an advertisement for soluble Pacific guano. Guano refers to bird droppings. And so they're selling you bags of fertilizer. And on the right we have one of the guano islands, which the United States took from Brazil because they wanted it for their farmers. Basically they passed a law and said these islands belong to us. Now, if you don't like that, Brazil, that's too bad. What are you going to do about it? So basically treating bird droppings in those days the way we treat oil these days. All right, let's look at the human renal system. Okay, so I mentioned the word filtrate. The word filtrate is the liquid that you get after you've pushed it through a filter. So that's what the word filtrate means. Okay, so the liver takes ammonia out of the blood and converts it into urea, which happens in the mitochondria of the liver, the hepatic cell mitochondria. And then it puts the urea back into the blood. The conversion of ammonia into urea is called the urea cycle, which is something that takes place inside mitochondrial cells, but in particular it takes place inside mitochondrial cells in the liver. And then the urea travels through the blood until it reaches the kidneys, where it is pushed through a little tiny microscopic filter called a glomerulus. The glomerulus is located inside a large filter unit that's called a nephron. Each individual kidney is composed of billions of nephridia that do this filtering, these little microscopic filters. And so the glomerulus, the glomerulus is located inside a structure called Bowman's capsule. The glomerulus pushes the blood serum out through the filter. The filtrate is then sent through a structure called the loop of Henle, where the water is taken back, as well as some of the salt. And then what's left is concentrated urine, which is sent to the center of the kidney through a structure called a collecting duct. Remember, the word duct means a small tube. And then from the center of the kidney, which is called the renal pelvis, it is transferred down to the urinary bladder and then out of the body. So what about the gross anatomy of the kidney system, the renal system? So we have a renal artery and a renal vein that take blood to the kidney and take blood away from the kidney respectively. Now you'll notice that there are two little glands on top of each kidney called the adrenal glands. They are not part of this process. They are classified, the adrenal glands are classified as part of the endocrine system. So you will see them on the diagram, but we're not going to worry about them here because they're not involved in the renal function. So we have two kidneys that filter the blood, filter the urea out of the blood. They do this through microscopic filters called nephridia, specifically thanks to a filter called the glomerulus. Right? And then the filtrate is sent to the renal pelvis, the center of the kidney, and then there's a smaller tube called a ureter that takes the urine from the renal pelvis to the urinary bladder, the bladder. And then from the bladder there's a single tube that goes out to the outside world called the urethra. So for both men and women, the little hole where the urine comes out is the end of a tube that is called the urethra. Right, so you need to know what the urethra is. We'll talk a little bit more about the urethra when we discuss the human reproductive system. All right, so here's the gross anatomy. You can see that there is a vein that takes blood from the kidney back to the blood vessel. Do you know which blood vessel this is? This is the big blood vessel that is taking blood from the lower part of the body back up to the bottom part of to the right atrium of the heart. Do you remember what it's called? Okay, and this artery on the right, so you have a large vein to the left and then an artery to the right. Sorry, it's reversed rather. So it's anatomically speaking, the large artery is on the left and the large vein is on the right. Okay, so they split off into two. So the large artery splits off into two smaller arteries that supply the left and right kidneys. So those are called the left and right renal arteries. And then the veins that bring it back to this large vein that's on the right-hand side of the anatomical body, they are the left and right renal veins. Okay, so what are these arteries and veins called again? Okay, so you probably guessed that the large vein is the inferior vena cava. So the large vein that runs up the center of the body, delivering blood from the lower part of the body to the heart, is the inferior vena cava. The large artery that takes blood downwards from the heart to the lower part of the body is called the abdominal aorta. So the left and right renal veins deliver blood to the inferior vena cava. The left and right renal arteries take blood to the kidneys from the abdominal aorta. That would make a good exam question, right? So make sure you know where they come from and where they go. Here's a cutaway image of a kidney. So on the outside of the kidney, we have a layer of connective tissue called the renal cortex, or it's actually, I apologize, the outer layer of the kidney is the renal cortex, and the renal cortex is surrounded on the very outside by a layer of connective tissue, which is called the renal fascia. The word fascia means face, right? So that it's the face of the kidney. So just under the renal fascia, which is sometimes also referred to as the renal cortex. Then we have not the renal cortex, but it's called the renal capsule. So we have a layer of connective tissue that covers the renal cortex. And that layer of connective tissue is called either the renal fascia or the renal capsule. So right under the renal capsule, the renal fascia, we have the renal cortex. Notice that the renal cortex is a lighter color than below. And then we have a bunch of, underneath that, we have a bunch of triangular shapes that are a darker color. Those are called, individually, those are referred to as renal pyramids because they're triangular shaped, they're pyramid shaped. And they have a darker color, and they form what is known as the renal medulla. So the inner part of the kidney is the renal medulla. Okay, and then, so the nephridia basically are long tubes that run up and down between the renal cortex and the renal medulla, and then when they're finished filtering, the urine is sent to the central part, this hollow part in the center. So there's a hollow part in the center of the kidney called the renal pelvis, where the urine collects. And if you look at it, if we were in the lab for this course, we actually have dissected animal kidneys so that you can look inside and see what it looks like. And you'll see that the central part of the kidney is yellow, has a yellow tinge to it. It has a yellow color, a yellow tint to it, because it's constantly holding this yellow urine. So urea is yellow in color. So the renal pelvis is yellow in color. It's a hollow spot in the middle of the kidney, and from there the urine is sent from the renal pelvis down a little tube to the bladder. This little tube is called a ureter. So we have a left ureter and a right ureter. So you can see that the left and right ureters go from the renal pelvis to the urinary bladder, and then there's a single tube that leads from the bladder to the outside world called the urethra. The urethra. And so don't confuse a ureter with a urethra. Those are two different tubes, but that would be a good exam question. Okay, so as I said, the outer part of the kidney is called the renal fascia, which means face in Latin, or the renal capsule. The renal fascia, also known as the renal capsule, are made of connective tissue, a tough layer of connective tissue. We always call the outer regions of an organ the cortex. So when we talk about the brain, we're talking about the cerebral cortex, and we'll talk about that later, but that's just an example. So you have the outer surface of an organ is usually referred to as the cortex. The inner part, the deep in the center part, is called the medulla. And so the renal cortex is light in color. The renal medulla is organized into a series of renal pyramids. And then at the very core of the kidney, we have the renal pelvis, which is where the urine is collected. The urine then flows down the ureter to the urinary bladder. So, once again, the structures of the gross anatomy of the kidney is we have a renal fascia, or a renal capsule, which is made of connective tissue on the surrounding the cortex of the kidney. Then we have the renal cortex, which appears lighter in color than the renal medulla. Then we have the renal medulla, which is the inside, which is made of a series of triangular shaped pyramids, which are called renal pyramids. The urine actually, once it's concentrated, ends up being collected in the hollow central part of the kidney called the renal pelvis. And then from the renal pelvis, the urine goes down the ureter to the urinary bladder. Now let's take a look at the nephron. The nephridia are the microscopic filters that actually do the filtration work. Okay, so a nephron is made of something called a Bowman's capsule. Right? A Bowman's capsule is a bulb-like structure that contains a little tiny capillary inside it called a glomerulus. Okay, now the glomerulus, it is a filter. So it filters the blood by pushing the blood serum, but not the cells, out into the Bowman's capsule. And then the filtrate, which is basically just blood serum that contains water, urea, amino acids, salts, a few other things. That's the filtrate. And so the red blood cells, the red and white blood cells, have not come out of the blood, just most of the serum. Most of the plasma. Most of the blood plasma is now the filtrate. Okay, now I said that then the filtrate goes down a little vessel and back up again, so that there's a vessel that travels. So the glomeruli of all the Bowman's capsules, so for every nephron, the Bowman's capsule and the glomerulus is located in the renal cortex. And then once the filtrate leaves Bowman's capsule, it goes down through a tube called the loop of Henle. It goes down into the renal medulla and then back up again. Now, the interesting thing is that within the cortex, the osmolarity of the renal cortex is 300 milliosmoles, which is the same as the blood. Down at the bottom of the loop of Henle, in the renal medulla, the osmolarity is 1,200 milliosmoles. So what do you think would happen to the filtrate, which contains a lot of water, if the filtrate goes down into the medulla the water will be sucked out by osmosis. And that's exactly how the water is reclaimed in the kidney. What is it that's causing the 1,200 millimolar milliosmoles of solute in the renal medulla? It's because the renal medulla is saturated with urea, the same way the tissue of a shark is, for instance. So the renal cortex has a low solute concentration, and when the filtrate travels through the loop of Henle down into the renal medulla, the water is sucked out of the loop of Henle, and then the filtrate goes back up into the top, up into the cortex again, and then down again through a larger tube, which is called the collecting duct. Sorry, the collecting duct. Right, and the collecting duct then travels once again down through the renal medulla, and sucks out some more water. This time, the collecting duct is a very special tube that contains some channels. We heard about cellular channels, right? So the epithelial tissues that surround the collecting duct are loaded with aquaporin channels that also help to remove the water and reclaim the water. And then the collecting duct empties what's left of the filtrate, which is now concentrated urine. It empties that concentrated urine into the renal pelvis. So let's take a closer look at all of this. Okay, so here on the left we have an image showing you where we're looking at. So we zoom in, we take a closer look at a nephron, which is located in the kidney. So here's the glomerulus, right there, the Bowman's capsule and the glomerulus. And this is located in the renal cortex, where the osmolarity is only 300 milliosmoles. So the glomerulus filters the plasma, the blood plasma, into the loop of Henle. The loop of Henle goes down into the renal medulla, where the osmolarity is 1,200 milliosmoles, and that's where water is taken out. And then it goes back up into the cortex again, and then back down again through another tube called the collecting duct. So over here on the right, we can take an even closer look at this. Okay, so here we have what's called an afferent arteriole. It's a small artery that takes the blood to the glomerulus inside Bowman's capsule. So the glomerulus is located inside this structure called Bowman's capsule. It operates as a filter. So this filter, the glomerulus, pushes the blood serum out, the liquid but not the cells, into the Bowman's capsule. The liquid then travels down through this structure here and back up again. This structure here is called the loop of Henle. Right? Now, because the loop of Henle is located down in the renal medulla, where the osmolarity is higher, the water is sucked out of the loop of Henle, and that water is picked up, it is absorbed by a network of veins that surrounds the loop of Henle that's called the vasa recta. So you do need to know what Bowman's capsule is, you need to know what the glomerulus is, you need to know what the loop of Henle is, and you need to know what the vasa recta is. Okay, so the vasa recta is a network of veins that surrounds the loop of Henle, and when the water is extracted from the concentrated urine, the filtrate that came from the Bowman's capsule, that water is absorbed by those veins and then put back into the general blood supply. Okay, so then the filtrate rather goes down the loop of Henle where the water is extracted, and then back up again, and then out through a second tube called the collecting duct. The collecting duct is able to extract even more of the water, to reclaim even more of the water, because the collecting duct is lined with a bunch of little pores. Each cell that lines the collecting duct has many channels in it that act like pores, and those channels are called aquaporin channels. The word aqua means water, porin means little pores, right? So we have the collecting duct, which is lined on the outside with these aquaporin channels. Okay, here's a better picture. Okay, so here's Bowman's capsule with the glomerulus inside. The glomerulus pushes the blood serum out into the Bowman's capsule. the filtrate, which is what, that's what the serum is after you've pushed it through a filter. It's a filtrate. So the serum then, the blood serum then travels down through the loop of Henle. At the bottom of the loop of Henle, some water is extracted and put into the vasa recta veins. Then it goes up through the loop of Henle and then out through the collecting duct, and even more water is collected on the way out of the collecting duct. Okay, let me show you that one more time here. So here we have the glomerulus inside the Bowman's capsule. It pushes the filtrate, it pushes the serum, blood serum out into the glomerulus. And then here we are in the renal cortex, where the osmolarity is 300 milliosmoles. The osmolarity of each layer of the kidney is listed here. Okay, so then the water travels down, the filtrate, the blood plasma travels downwards into the renal cortex. Into the renal medulla. I'm getting quite muddled here. Travels down into the renal medulla, and as it travels down to the bottom where the osmolarity is 1,200 millimoles, the water is sucked out. And then it goes back up again, and then it travels out through the collecting duct. Okay, so on the way back up the loop of Henle, we extract more water, but we also extract some of the salt on the way back up, including urea, which is a salt. And so most of that salt is put into the renal medulla because that's where we need to have a high osmolarity in order to make this work. All right, and then we go up the loop of Henle and then down the collecting duct where even more water and more salt is taken out, especially urea. The urea is put into this area in the renal medulla. Now I mentioned that the collecting duct that sends the urine down to the renal pelvis. It has aquaporin channels in it, and it's these aquaporin channels that are able to extract the water from the filtrate. Now the number of aquaporin channels, and this is a critical point, you need to know this, the number of aquaporin channels is regulated by a hormone called vasopressin, also known as antidiuretic hormone. Right? So the number of aquaporin channels that are put into the collecting duct is regulated by a hormone called antidiuretic hormone. And so antidiuretic hormone is made in a structure in the brain called the pituitary gland. We'll talk more about the pituitary gland when we talk about the endocrine system. But the pituitary gland produces antidiuretic hormone. The more antidiuretic hormone that the pituitary gland of the brain produces, the more aquaporin channels are put in to the collecting ducts of the nephridia in the kidneys, and the more water is extracted from the urine. So therefore it stands to reason, if your brain produces a lot of aquaporin, the collecting ducts of each nephridia will have lots of aquaporin channels in them, and they will extract a lot of water from the filtrate, and then you will urinate very little. And the urine that comes out will be very concentrated. It will be mostly urea with very little water in it. On the other hand, if the brain produces less antidiuretic hormone, the collecting ducts of each of the nephridia will have fewer aquaporin channels and will reclaim less of the water, so you'll urinate more often, and the urine that comes out will be more dilute. So you can see that the amount of water that is retained by the kidneys is regulated or controlled by the brain, by the pituitary gland specifically, which is part of the endocrine system that's located near the brain. Okay, so the amount that we urinate is controlled in part by the amount of antidiuretic hormone that's produced by the pituitary gland near the brain. Okay, so hormonal control of urination. The word diuresis means excessive urination, right? So antidiuretic hormone is a hormone that acts against diuresis. So diuresis means excessive urination, and antidiuretic hormone means something that fights against diuresis. So antidiuretic hormone is produced by the pituitary gland, which is located at the base of the brain, Antidiuretic hormone is also known as vasopressin. And now, what does the pituitary gland use to tell whether it needs to produce more antidiuretic hormone or not? Okay, this is important because how does the brain know? How does the pituitary gland know how much antidiuretic hormone to produce? The pituitary gland is going to produce more antidiuretic hormone if it perceives that you are losing too much water. If the brain thinks that you're losing too much water, it will tell the pituitary gland to produce more antidiuretic hormone, to produce more vasopressin. So how does the brain tell that you're losing too much water? How does it tell? Now, it just so happens that there are receptors in the brain that are called osmoreceptors that are able to sense how salty the blood is. There are receptors in the brain, they're actually neurons, specialized neurons, that are able to tell the concentration or the osmolarity of the blood. If the blood has a high osmolarity, the brain assumes that you're losing water through perspiration. Okay, so imagine this. If you had, let's say you're in chemistry class, and let's say that you have a beaker of water, you have a 1 liter beaker of water, and you dissolve 1 mole of sodium chloride into that beaker, right? What's the concentration of that solution? All right, if you said that the concentration of that, I dissolved 1 mole of sodium chloride in 1 liter of water, so that would give me a 1 molar solution of sodium chloride, wouldn't it? Okay, now what if I put that beaker on top of a heating plate and I evaporate 500 mils of the water out of that beaker? The salt is going to stay in the water, and the water will evaporate, right? So I will have left the salt, the amount of salt in that solution, the amount of salt in that beaker will remain the same, but I've removed half the water. Okay, so I originally put 1 mole of salt into 1 liter of water, and that gave me a 1 molar solution of sodium chloride. I then removed half the water. So now I have 1 mole of salt dissolved in 500 milliliters of water. What's the concentration of that solution? Right? Can you tell me the answer to that? If I removed half the water, so I now have 500 mils of water instead of 1,000, but I have the same amount of salt, 1 mole. 1 mole dissolved in half a liter is going to be 2 molar, isn't it? It's going to be a 2 molar solution. So if you keep the amount of salt in a solution the same, but you remove half the water, the concentration, the effective concentration goes up. The volume goes down. Yes, certainly the volume goes down, but the concentration goes up. Okay, so the brain, when the brain senses that the concentration of salt in the blood has increased, the brain assumes that that's because the volume of blood has decreased. The amount of water in the blood has decreased, usually because you were either urinating too much or you were perspiring too much. And so the brain says, oh my goodness, the saltiness of the blood has increased. It must be because you're losing water. So I'm going to tell the pituitary gland to produce antidiuretic hormone that puts more aquaporin channels into the collecting ducts of the nephridia, and you will retain more water until the salt concentration in your blood goes back down because the volume of water in your blood has gone up. That's what the brain thinks. That's what the brain assumes. It may not be correct. Right? That may not actually be the reason. So the reason why the salt concentration in your blood has gone up may be for another reason. The concentration of salt in your blood may not have gone up because you lost water. The concentration of salt in your blood might have gone up because you ate a bunch of salty food. Nevertheless, the brain doesn't realize that because the brain is simply judging how much water that you're losing by measuring the concentration of salts in your blood. And the concentration of salts in your blood might have gone up simply because you ate salty food. Okay, so what happens is that if the salt concentration of your blood has gone up because you ate salty food, it means that the volume of blood did not go down. But if the kidney suddenly starts retaining more water, in fact, the volume of your blood will go up even though it was never low in the first place. So what happens is, what do you think happens to the blood pressure when you increase the amount of water in the blood? What do you think happens to the pressure? Right? The blood pressure will go up if the volume goes up. Right? So what if we have a hose and we suddenly put twice as much water into that hose as we had before without increasing the size of the hose? So we don't increase the size of the blood vessels. We don't do vasodilation. So we don't increase the volume of the blood. We don't increase the size of the blood vessels, but we increase the amount of water that's in those blood pressure in those blood vessels, and that causes an increase in blood pressure. Right? So if you eat salty food, your blood pressure will go up for the simple reason that your kidneys will start retaining more water, and therefore your blood volume will go up. If your blood volume goes up, the blood pressure goes up. Now, I've just taught you one little example of how having salty food or increasing the amount of salt in your blood results in your retaining more water in the kidneys and causing your blood pressure to go up. If you take Biology 130, I'll teach you about this system again, the antidiuretic hormone system, which causes the blood pressure to increase. But I'll also teach you about a couple of other hormones that either cause the blood pressure to increase or to decrease. So in fact, the blood pressure in humans is carefully controlled by several factors. So there's several different hormones, some of which work against each other. You know, the way we learned about muscles, you have antagonists and you have agonists and antagonists, where you have a set of muscles that work in opposite directions. The same is true for many of the hormones in the human body. There's some hormones that cause your blood pressure to go up. There's some hormones that cause your blood pressure to go down. And the blood pressure is regulated homeostatically through homeostasis. And so all of these factors are carefully controlled, and when you're young, all of those systems that control the blood pressure work perfectly. As you get older, they don't work as efficiently, and so older people tend to have odd blood pressure, usually blood pressure that's too high. Blood pressure that's too high is called hypertension. Most people that are, over 40, over 50, certainly over 60, most people that are that age have chronic hypertension, meaning they have long-term high blood pressure. And that's because there's so many things that control the blood pressure. It's very difficult to keep them all balanced and coordinated as your body is starting to wear out. Okay, so antidiuretic hormone causes us to put more aquaporin channels into the collecting ducts of the nephridia, so you retain more water. So this is a diagram showing homeostatic control of blood pressure by antidiuretic hormone. Okay, so we have a stimulus, which is, remember that for homeostasis, you have a stimulus and then you have either positive or negative feedback. The stimulus is when one of these parameters of the body deviates from the normal range. So the normal osmolarity of blood is supposed to be, human blood is supposed to be around 300 milliosmoles. So if the concentration of solutes in the blood increases, let's say it goes up to 350 milliosmoles, that's a stimulus. That is considered to be a stimulus. And then your body does something, responds to that with what's called negative feedback. Negative feedback means the body does something to bring whatever it is you're looking at back into the normal range. So if the osmolarity of human blood is normally 300 milliosmoles, and if it suddenly goes up to 350 milliosmoles, that's a stimulus. What does the body do as a negative feedback to bring it back down to 300 milliosmoles? Okay, so what happens is there are special receptors in the brain. It's a structure in the brain called the hypothalamus, by the way, and it contains receptors that are able to sense how salty the blood is. Those receptors are called osmoreceptors. They are specialized neurons that are able to sense how salty the blood is. So the osmoreceptors suddenly sense that the blood is too salty. The brain assumes that the blood has gotten saltier because you've lost water. So we lose water, but keep the, there are two ways that the amount of salt that's dissolved in the blood can go up. One way is you could put more salt into it, The other way is you could remove some of the water. Either way, the concentration of salt in the blood is going to go up. Okay, so the brain, there are two ways we can cause this, but the brain only understands one of those ways. The brain doesn't know anything about us eating pizza or chips and things that are loaded with salt. So the brain doesn't assume that. The brain assumes that our blood has gotten more salty because we've lost water, and it says, I have to do something to retain more water. So these osmoreceptors sense that the saltiness of the blood has increased, and it causes, on the one hand, it causes the pituitary gland of the brain to produce more antidiuretic hormone, which in turn puts more aquaporin channels into the collecting duct. And at the same time, the brain also makes us thirsty. So we go around drinking more water. So that's why when you eat salty foods, you get thirsty. And when you eat salty foods, you retain more water, which makes your blood pressure go up, and you urinate less if you eat salt. So you've seen people, like, they go out in the desert and they take salt pills to avoid urinating so much because they want, you're losing so much. If you're out in a hot desert, you can take salt pills to retain water because you're losing so much water through perspiration. You're losing a lot of water through the skin. So the least you can do to counteract that is you can take salt tablets that make you urinate less, because they cause those salt tablets cause your brain to produce more antidiuretic hormone. Okay, so I could ask you to explain why and how salt controls blood pressure, or how increases in salt lead to increases in blood pressure. I could ask you that question on a test. I could ask you to write that out as a very short essay or something like that. All right, so we also, the other thing the brain does in the face of that kind of a stimulus is it makes you thirsty. So you drink more water, which then brings the salt you increase the amount of water in your blood. You have not increased the size of the blood vessels, so the blood pressure did go up, but you did add more water to the blood, number one, by drinking, and number two, by retaining more of it in the kidneys. Okay, so this is how osmoregulation of the blood pressure, which involves antidiuretic hormone, works. But as I said, there are other hormones that act in the opposite direction. There's some other hormones that cause you to lose water because your blood pressure is too high, and we won't talk about those in this course. However, now you know an interesting thing, which is why, maybe some of you, I know that many of you don't drink alcohol, you don't go to pubs and bars and drink beer, and good for you. But if you do go to pubs and drink beer, you'll notice that in bars and pubs they give you free peanuts, free salted peanuts and free salted pretzels. And they're not doing that because they're generous. They're doing that because they want you to eat salty food so that you'll be thirsty, and then you'll buy more beer and drink it. And so that's exactly why bars and pubs serve salty things like salted peanuts and salted pretzels, because they're exploiting the fact. Now, I don't think that there are many bartenders that realize the reason why the free peanuts make everybody thirsty is because the pituitary gland produces more antidiuretic hormone, which puts more aquaporin channels into the collecting ducts of each nephron. I don't think there are too many bartenders that realize that. There might be some, but now you know the theory behind why that works. Okay, so to summarize what we talked about, we mentioned the fact that the original purpose of the renal system was to regulate the amount of water inside sea animals like fish that live in the ocean. The second purpose of the renal system was to get rid of nitrogenous wastes like ammonia that results from the breakdown of amino acids and nucleic acids. And so for humans who live on land, we don't live in the ocean anymore, the osmoregulation part of the osmoregulation function of the renal system is less important than the filtration of ammonia out of the bloodstream. We learned about the gross anatomy of the kidneys. We talked about the renal fascia and the renal cortex and the renal medulla and the renal pelvis, the ureters and the urea and the urinary bladder. And then we learned about the microanatomy of the renal system, which involves the anatomy of nephridia, a nephron. A nephron is a microscopic filter that you can't really see without a microscope. It's not quite big enough to be seen without a microscope. It's pretty big actually, but just not big enough to see without the help of a microscope. So we talked about the structure of an individual nephron, which includes the glomerulus, which is a little tiny capillary that filters the plasma out of the blood into Bowman's capsule. From Bowman's capsule, it travels down into the renal medulla and back up again through a structure called the loop of Henle. The water that's extracted from the filtrate, the blood plasma, as it goes down into the bottom of the loop of Henle, is sucked out of the loop of Henle and put into some veins called the vasa recta, and those veins are ultimately attached to the renal vein that puts the water back into the bloodstream. So the water that's recovered gets put back into the bloodstream because it was absorbed by the veins that constitute the vasa recta. The vasa recta are the veins that surround the loop of Henle. Okay, and then the filtrate is now much more concentrated, but it goes down through the collecting duct to the renal pelvis. On the journey through the collecting duct, even more water is extracted back out, even more water is reclaimed. And the amount of water that is reclaimed as the urine goes down the collecting duct is determined by the number of aquaporin channels that are in the collecting duct. And the number of aquaporin channels that are in the collecting duct is determined by the amount of antidiuretic hormone, also known as vasopressin, that the pituitary gland is producing. The pituitary gland is producing antidiuretic hormone under the control of the hypothalamus, which is a part of the brain. The hypothalamus has the ability to sense how salty the blood is, and it will increase the production of antidiuretic hormone if the concentration of salt in the blood becomes too high. And so the brain is assuming that the concentration of solutes in the blood has increased because you've lost water, but that may not be true. It may simply be true that we ate a lot of salty food, and that's why the concentration of salt in the blood went up. And that's why it's a good idea not to eat salty food, because eating salty food increases blood pressure. The blood pressure increases because we've added more water to it by extracting more water from the urine in the collecting ducts. So eating salty food causes you to retain water in the bloodstream, and that causes an increase in blood pressure. Right? So there are several ways that you can increase the blood pressure. One way is that you can increase the pulse rate. So if the heart rate goes up, you're pumping the blood faster through the blood vessels, and that causes an increase in pressure. Another way that you can increase the blood pressure is simply to increase the volume of blood that is in the same blood vessels. So we haven't increased the number of blood vessels and we haven't increased the size of the blood vessels, but if we add water to the blood, that will increase the volume of blood that is flowing through the same blood vessels, and that's another way to increase the blood pressure. Okay, so we're not really going to discuss it in this course, but how do we lower the blood pressure again? One way is to dilate the blood vessels, vasodilation. So you relax the smooth muscles that are surrounding the lumen of the blood vessel, so that can lower the pressure. But you can also the brain can also produce other hormones, and the heart itself actually produces some hormones that if the heart is having to work too hard because the blood pressure is too high, there is another hormone that's produced by the atria of the heart that causes us to urinate more. So while vasopressin, antidiuretic hormone, is causing us to urinate less, there's another hormone that's being produced at the same time that causes us to urinate more to get rid of the water. These two hormones are in constant balance. There's a third hormone called angiotensin that causes us to urinate less. And so there's basically two hormones that make us urinate less and retain water, versus one hormone that causes us to urinate more and lose water. And that balance of hormones is in perfect alignment when we're young, where the blood pressure is normal, typically 120/70, something like that. As you get older, it gets harder and harder to coordinate those factors, and so most elderly people, certainly people over 40 or 50, have chronic hypertension. and so they have to be more careful to not eat salty foods because you don't want to have higher blood pressure. If you have higher blood pressure, there's a number of things that can go wrong. All right, so the next lecture, this is our vocabulary. There's one word here, malphagian tubules. I wouldn't worry about that. That has to do with something that I didn't teach you, so don't worry about the malphagian tubules. But everything else you can look up in the vocabulary. So the next lecture is on the human reproductive system, and I will see you there.